Hello everyone and welcome to Ghar's YouTube channel. Today we shall discuss the 2020 South Asian University entrance paper. South Asian University is an international university sponsored by the South Asian Association of Regional Cooperation. It conducts this entrance exam for MSc Biotechnology course admissions. The paper has 100 questions and is divided into two sections. Part A has 30 questions and Part B has 70 questions. Please note that all the questions in the paper that we are discussing today are entirely memory based as the complete paper has not been released by the institute. So let's move on to the first question. Here the first question is in the form of a circuit diagram and we are supposed to find the current flowing through the circuit. Here is the solution. We can see that the first two resistors are in series. So their net resistance is R plus R. Since the value of both these resistance is the same, so that is 2R. Similarly, the next two are also in series. So we will name the net value as R net 2, which is also equal to R plus R, that is 2R. Now, R net 1 and R net 2 are in parallel. So, 1 by R equivalent is equal to 1 by R net 1 plus 1 by R net 2, which is equal to 1 by 2R plus 1 by 2R. So R equivalent becomes 2R by 2, which is equal to R. Now we know by Ohm's law that V is equal to IR. So I is equal to V by R, since the R equivalent here is R. Find the value of sine inverse 1 by root 2. Now, we should let the value be theta. So to find theta, theta is equal to sine inverse 1 by root 2. Sine theta is therefore 1 by root 2. We know that sine 45 is 1 by root 2. So theta is equal to 45 degree. Serotonin is made up of which amino acid? Serotonin is made up of tryptophan. The synthesis of serotonin occurs by this pathway. Tryptophan is converted into 5-hydroxytryptophan, that is 5-HTP, by tryptophan hydroxylase. 5-HTP is acted upon by 5-HTP decarboxylase to form 5-hydroxytryptamine, that is serotonin, which is 5-HT. This amino acid, that is tryptophan, must be entered in our body through diet, and it is commonly found in foods such as nuts, cheese and red meat. Thus, tryptophan deficiency can lead to lower serotonin levels. Crossing over occurs at which stage in meiosis 1? Crossing over or recombination occurs during the Pachytene stage of meiosis 1. Pachytene stage occurs during prophase 1. Recombination involves mutual exchange of genetic material, that is of the corresponding segments of the non-sister chromatids of homologous chromosomes, and produce variations. It takes place by breakage and reunion of these chromatid segments. As you can see here, in the prophase 1, there is crossing over or recombination happening. This occurs during Pachytene stage of prophase 1 in meiosis 1. Here, the next question asks about the number of chromosomes and chromatids at metaphase 1 of meiosis. Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. Now, if we assume the original genetic information to be 2n, since the organism is diploid, we will find the following results. First, we will look at the steps. Meiosis 1 has interphase 1, prophase 1, metaphase 1 and anaphase 1. In interphase, the chromosomes duplicate. In prophase 1, the homologous chromosomes pair and exchange the segments, that is recombination. In metaphase 1, these tetrads of homologous chromosomes line up on the equatorial plane. In anaphase, the sister chromatids remain attached, but the pairs of homologous chromosomes split up to the ends of the spindle fibers. Now here is the number of chromosomes. If we assume the initial number of chromosomes to be 2n at the beginning of prophase 1, the number of chromosomes is 2n and the number of chromatids is 4n, since one chromosome has 2 chromatids. So in metaphase 1 also, the number of chromosomes remains 2n and the number of chromatids is 4n. Since this is the only time there are tetrads present, we call this n number of tetrads. So, in short, the number of chromosomes and chromatids at metaphase 1 of meiosis is 2n and 4n respectively. 200 gram flour and 25 gram of fat is required to make a cake. How many cakes can be made with 5 kg flour and 1 kg fat? Okay. Now, cakes that can be made using 5 kg flour if we require 200 grams of flour for one cake. 5 kg is equal to 5000 grams. So 5000 divided by 200 per cake is equal to 25. So we can make 25 cakes with this amount of flour. Cakes which can be made with 1 kg fat. 1 kg fat again is equal to 1000 gram of fat. Each cake requires 25 gram of fat. So 1000 by 25 is equal to 40. So now we have enough flour to make 25 cakes and enough fat to make 40 cakes. So we can make 25 complete cakes. A bag contains 6 white and 9 black balls. Find the probability of drawing a black ball. Okay, let us first assume that the 6 white balls are numbered from W1 to W6 and the 9 black balls are numbered from B1 to B9. So the sample set will contain W1 through W6 and B1 to B9. So the number of elements in the sample set is 15. Let A be an event of drawing a black ball. So the set A will contain B1 through B9 since we are drawing only one ball. So the number of elements in A, it will be 9. The probability of event A will be P equal to number of elements in A divided by number of elements in S, which is the sample set, which is equal to 9 by 15. So the probability of drawing a black ball is 9 by 15. The next question asks about the energy stored in a capacitor. The energy U stored in a capacitor is electrostatic potential energy and is thus related to the charge Q and voltage V between the capacitor plates. A charged capacitor stores energy in the electrical field between its plates. This energy is denoted by eta V, which is equal to half epsilon E square AD, which is equal to half epsilon V square by D square AD, which is equal to half epsilon A by D into V square, which is equal to half CV square, where C is the capacitance and V is the voltage. In de Broglie wavelength, which constant is used? De Broglie wavelength is usually represented by the symbol lambda. For a particle with momentum p, which is also denoted as mv, the de Broglie wavelength is defined as lambda is equal to h divided by momentum, that is p or mv. Here h is the Planck's constant. The Planck's constant says how much energy of a photon increases when the frequency of its electromagnetic wave increases by 1 in SI units. It is named after the physicist Max Planck. In SI units, the Planck's constant is exactly 6.626 into 10 raised to minus 34 meter square kg per second. Mean equal to median equal to mode is a condition of which function? The conditions that determine the relationships between mean, median and mode, that is the three central tendencies, are as follows: negatively skewed, positively skewed, or normal, that is no skewed. Normal distributions are symmetric around their mean. The area under the normal curve is equal to 1. These normal distributions are denser in the center and less dense in the tails. They are defined by two parameters, that is the mean and standard deviation. It represents a perfectly symmetrical distribution. Hence, mean, median and mode lie on the same point in the curve. So, mean is equal to median is equal to mode is a condition of normal distribution function. Which of the following are quantitative functions? A. Seed color, B. Flower color, C. Yield, or D. Seed protein content. Quantitative functions can be measured in terms of SI units. Out of these options, seed protein content is the only one which can be measured in terms of concentration. So, the correct answer is option D, that is seed protein content. Erythropoietin is released from. Erythropoietin or EPO is a hormone produced primarily by kidneys with small amounts made by the liver. It acts on red blood cells to protect them against destruction and also stimulates stem cells of the bone marrow to increase the production of red blood cells. These red blood cells carry oxygen from the lungs to the rest of the body. Approximately 90% of systemic EPO in adults is produced by peritubular interstitial fibroblasts in the renal cortex and outer medulla of the kidney, and it is released through renal canal. So this is how erythropoietin affects RBC concentration, and it is released by kidney. Gluconeogenesis occurs in. Gluconeogenesis is defined as the de novo synthesis of glucose from non-hexose precursors. This is a diagram of gluconeogenesis. As you can see here, different non-hexose precursors such as glycerol, which is a component of fatty acids, amino acids, acetone, lactate, etc., enter the pathway to ultimately form glucose. The major site of this process is the liver, with small amounts also taking place in the kidney. Even little gluconeogenesis takes place in the brain, skeletal muscle or heart muscle. But the primary center is the liver. If allele capital C, that is full shaped pod, is dominant over small c, that is constricted, capital G, that is yellow, is dominant over small g, that is green, then the genotype of yellow and constricted is. Okay. Yellow, that is capital G, is a dominant trait. So it will always express itself even in heterozygous form. Thus, both capital G and capital G small g will show yellow pod phenotype. Now, the other trait, that is constricted, is a recessive trait. So, it is only able to express in homozygous conditions. So, only small c will show a constricted pod. Thus, both capital G small c or capital G small c genotypes will show yellow and constricted phenotype. If we add 5 with each term, then which of the following will not change? A mean, B median, C standard deviation, or D all of the above. As a rule, the median, mean and quartiles will be changed by adding a constant to each value. However, the range, interquartile range, standard deviation and variance will remain the same. So, the correct answer is option C. Additionally, if we multiply to each term by a constant value, it will multiply the mean, median, quartiles, range, interquartile range as well as standard deviation by that constant. And for variance, it will multiply the variance, which is simply the square of standard deviation, by square of that constant. T helper cell is identified by: the T helper cell is a type of T cell that provides help to other cells in the immune response by recognizing foreign antigens and secreting substances or cytokines that will activate T and B cells. These cells have CD4 markers on their surface. They are thus a special subpopulation of CD4 cells. As you can see, we can easily distinguish a TH cell, that is a T helper cell, by a cytotoxic T cell with the help of CD4 markers. So that's how you identify a T helper cell. A T helper cell will contain A. TNF alpha, CD3 plus, CD4 plus, CD8 minus, B. TNF alpha, CD3 plus, CD4 minus, CD8 minus, C. TNF alpha, CD3 minus, CD4 plus and CD8 plus. For this, we shall first refer to this diagram. This is again a compare between TH cells and TC cells. Now, TH cells or T helper cells have CD3 and CD4 markers on their surface, while CD3 markers are not unique to T helper cells since they are also found here on TC cells. CD4 markers, however, are extremely unique to TH cells. Cytotoxic T cells, that is TC cells, have unique CD8 markers for their recognition. So, from the options, we have to find CD4 plus and CD3 plus, which is the first option. And it does not have CD8, so CD8 minus. Now, TNF alpha, that is tumor necrosis factor alpha, is an inflammatory cytokine which is produced by activated macrophages, T lymphocytes and natural killer cells, that is NK cells. So obviously it has TNF alpha, CD3 plus, CD4 plus and no CD8, that is CD8 minus. So the correct answer is option A. Hypersensitivity requires which immunoglobulin? For this, we shall refer this diagram. Now, as we can see, type 1 hypersensitivity reactions involve IgE antibody against soluble antigens, triggering mast cell degranulation. This is within 1 hour, and examples are anaphylaxis. Type 2 hypersensitivity reactions involve IgG and IgM antibodies, that is immunoglobulins, against cellular antigens, leading to cell damage mediated by other immune system effectors. Type 3 hypersensitivity reactions involve interactions of IgG, IgM and occasionally IgA1 antibodies with an antigen to form immune complexes. Accumulation of these immune complexes in tissue leads to tissue damage mediated by other immune system effectors. Type 4 hypersensitivity is T cell mediated. The examples of each of these are anaphylaxis for type 1, hemolytic anemia for type 2, SLE for type 3, and rash or SJS for type 4. Phenylalanine is produced by which pathway? The pathway involved in the production of phenylalanine is called as the Shikimate Pathway. This pathway links metabolism of carbohydrates to biosynthesis of aromatic compounds. In a sequence of 7 metabolic steps, phosphoenol pyruvate and erythrose 4-phosphate are converted to chorismate, the precursor of amino acids, and many aromatic secondary metabolites. In higher plants, many aromatic compounds are derived from phenylalanine, tyrosine, and tryptophan, which are the end products of shikimic acid pathway. The next question talks about wavelengths required for photosynthesis. Here is a diagram to understand better. The wavelengths of visible light for photosynthesis fall within the ranges 425 to 450 nanometers, that is the blue range. As you can see here, the absorption spectrum is quite high in this range, and 600 to 700 nanometers, which is called as the red range. And here is also another peak in the absorption spectrum. Therefore, the best light sources for photosynthesis should ideally emit light in blue and red ranges. So plants absorb blue and red light for photosynthesis. As you can see here, chlorophyll A, chlorophyll B, and beta carotene, which are important pigments for these absorption, also show a spike in these regions, that is the blue and the red regions. So the correct answer is blue and red light. Light reaction occurs in. Okay, so basically what is the light reaction? When the light hits chlorophyll A, it gets excited to higher energy state, followed by a series of reactions. This energy is then converted into energy stored molecules such as ATP and NADPH using PS1 and PS2. So you can see here it is stored in NADPH. This entire process occurs in thylakoids of the chloroplast, which are these individual stacks. Dark reactions, on the other hand, which are also known as carbon assimilation reactions, take place in stroma. Which rays are emitted by the nucleus? To understand the different types of radiation, we will look at this diagram. So we have radio waves, we have microwaves, we have infrared, we have X-rays and gamma rays. In the question, they have asked which rays are emitted by the nucleus. Energy given off by matter in the form of tiny, fast-moving particles such as alpha particles, beta particles, and neutrons, or pulsating electromagnetic rays or waves such as gamma rays, are emitted from nuclei of unstable radioactive atoms. So here the answer would be gamma rays, alpha rays, or beta rays. When talking about radio waves, naturally occurring radio waves are emitted by lightning and astronomical objects as a part of blackbody radiation. They are generated artificially by transmitters and received by radio receivers using antennas. Microwaves. For these kind of waves, cosmic microwave background radiation is a natural source of microwaves. They are used to study the Big Bang. Stars, such as the sun, are also responsible for natural emission of microwave. Under the right condition, even atoms and molecules are able to emit microwaves. Inside an oven, they are produced by an electron tube called magnetron. These microwaves are reflected within the metal interior of the oven and are absorbed by the food, which helps to cook food better. Now, talking about infrared rays, the primary source of infrared radiation is heat or thermal radiation. So, any object which has temperature radiates in the infrared. So, this was just some additional information. The next question asks us to choose the correct option with the correct ploidy, that is haploid, diploid and triploid. First, egg, nucellus, endosperm. B, egg, endosperm, nucellus. Or C, integument, egg and nucellus. For this, we shall refer to these two diagrams. First, the egg cell, that is this cell, is haploid, having half the number of chromosomes of all the cells in the plant body, which are diploid cells. Now, nucellus here is the central part of the ovule containing the embryo sac. It contains reserved food materials and is thus diploid. Now, each ovule receives a pollen tube that delivers two sperm cells to the embryo sac. One sperm fertilizes immediately with the egg, generating the diploid zygote, while the other sperm fertilizes with the central cell, giving rise to the endosperm that is usually triploid. It is triploid because these two polar nuclei, which are haploid each, fuse with the haploid sperm. So the total ploidy becomes triploid, that is 3n. So the correct answer is option A. Egg is haploid, nucellus is diploid and endosperm is triploid. What is a pollen chamber? For this, we will refer to this diagram. As you can see, the pollen chamber is correctly shown here. This is the pollen chamber. This is the nucellus, at the tip of which we have the pollen chamber. So the pollen chamber refers to a small chamber at the apex of the nucellus in some plants, which is mostly gymnosperms, for the reception of pollen. In Bt cotton, what is Bt? Bt stands for naturally occurring bacterium Bacillus thuringiensis. Bacillus thuringiensis lives in the soil and is found all over the world. Some types of Bt produce a protein crystal that is toxic to insects. So this is the crystal. These protein crystals have been used in organic farming for over 50 years to control insects. The genes producing these proteins are now engineered in plants so that they can make the protein in their own cells. On ingestion of these crystals by a larva, that is insect larva, the crystals are first solubilized to form protoxin. When this protoxin reaches the alkaline pH of the gut, it is converted into the activated toxin, which binds to the receptors to increase the permeability of the gut wall. This eventually allows free flow of substances through the gut wall, and due to lysis of the cells, the larva dies. This is the function as to why Bt cotton, that is Bt, that is in general Bacillus thuringiensis, is used as a biopesticide. The next question talks about the common secondary messengers. Option A, CAMP. Option B, tRNA. Option C, GTP. Second messengers are intracellular signaling molecules released by the cell in response to exposure to extracellular signaling molecules, that is, the first messengers. The two most commonly and widely used secondary messengers are cyclic AMP, that is CAMP, along with calcium ions, that is Ca2+. A large variety of relay proteins are sensitive to cytosolic concentration of one or the other of these secondary messengers. So the correct answer is option A, CAMP. The next question talks about the first plant produced, which is virus resistant. The answer to this is Nicotiana tabacum. The expression of coat protein gene of tobacco mosaic virus in transgenic tobacco, that is Nicotiana tabacum plants, is the first example of transgene mediated resistance to a plant virus. So the correct answer is tobacco. Aedes mosquito is a vector for Aedes aegypti, which is a well-known species of mosquito known for their striped nature, is a vector for several viruses including yellow fever virus, dengue virus, chikungunya virus, along with Zika virus. So it can lead to the spread of yellow fever, dengue, chikungunya and Zika. A student will perform t-test for. A t-test is a type of inferential statistic used to determine if there is a significant difference between the means of two groups, which may be related in certain features. The t-test is one of the many tests used for the purpose of hypothesis testing in statistics. A t-test is also of different types, so to determine which t-test to use, here are some key features. The student can first determine if the sample sets, that is the two sets that he or she wants to test the hypothesis, are same or related. If yes, they use the paired or dependent t-test. If not, they move on to the next question, which is, are the two sample sets of the same size? If they are, we use equal variance independent t-test, and if they are not, we check for if the samples have the same variances. If yes, we still use the equal variance independent t-test. If not, we use the unequal variance independent t-test. But the t-test will always be performed between means of two groups. Always use two sample sets. Which disease is caused by defective nucleotide excision repair mechanism? This is a diagram of the nucleotide excision repair mechanism. It is the main pathway used by mammals to remove bulky DNA lesions such as those formed due to UV light or environmental mutagens and some other chemotherapeutic adducts from the DNA. Deficiencies in NER, that is nucleotide excision repair, are associated with extreme skin cancer prone inherited disorder such as xenoderma pigmentosa. So a very famous disease caused due to defect in nucleotide excision repair mechanism is xenoderma pigmentosa. When a cell has an ability to produce all differentiated cells, it is known as. Okay, so to understand the different terms when it comes to potency, we have this diagram here. Okay, so from a oocyte, when during a morula stage, we call the cells to be totipotent. That is, each cell can develop into a new individual. After the formation of blastocyst, when the inner mast cells are formed, along with trophoblast cells, but the inner mast cells in particular, which give rise to further germ layers such as the endoderm, mesoderm and ectoderm, they are called as pluripotent. Pluripotent means each cell can develop into over 200 different types of cells. Now, each germ layer, that is endoderm, mesoderm or ectoderm, these initial cells are known as multipotent cells. So these cells can differentiate and form a number of tissue types. So these can form endodermal cells, can form endodermal organs such as the respiratory system. Mesoderm gives rise to circulatory system and ectoderm gives rise to nervous system. Once these cells are formed in the systems, they are called as unipotent cells. So the potent cells in these organs are unipotent, that is, they can differentiate along only one lineage. So, going back to our question, when a cell has an ability to produce all differentiated cells, it is known as totipotent, where each cell can develop into a new individual to form all different differentiated cells. So the correct answer is totipotent. The cell is known to be totipotent if it can differentiate into all cells. Okay, the next question talks about the basic principle of Southern blotting. A Southern blot is a laboratory method used to detect specific DNA molecules from among a variety of other DNA molecules. The technique was named after its inventor, that is Edward Southern. Here is a diagram to understand better. Southern blotting is based on the principle of separation of DNA fragments by gel electrophoresis, as shown here, followed by the identification by labeled probe hybridization. The DNA fragments are separated based on their size and charge during electrophoresis. The technique involves the transfer of electrophoresed DNA from electrophoresis gel to a nitrocellulose membrane. The nitrocellulose filter is then sandwiched between the gel and a stack of paper towels, which draws the transfer buffer from the gel through capillary action. The desired DNA is then detected once transferred onto the filter with the help of labeled probe, which is complementary to the desired DNA. So this is the basic principle of Southern blotting. The next question asks the examples of CAM plants. CAM metabolism, that is crassulacean acid metabolism, is common in plants that live in hot and dry environments where water is difficult to gain and conserve. During night, CO2 is fixed into malate, which is stored as malic acid in the vacuole. During the day, when sunlight is present, this malate enters chloroplast, where it enters into Calvin's cycle. The examples of CAM plants include the species of cacti such as euphorbia, agave, as well as Clusia pratensis. Other facultative CAM plants that can switch between the two modes of carbon fixation include Calandrinia polyandra, Mesembryanthemum crystallinum, Portulaca oleracea, and Talinum triangularum. If oxygen is 2% less, then A. C3 is more efficient than C4, B. C4 is more efficient than C3, C. No effect, or D. Efficiency of both C3 and C4 increases. If the percentage of oxygen in the air is reduced, rubisco, will function as a carboxylase. So, C4 plants have a better CO2 use efficiency since there is no photorespiration. Thus, C4 plants will be more efficient than C3. However, the efficiency of both C3 and C4 will increase as rubisco functions as carboxylase. In protoplast fusion, which of the following compounds is used? A. Sorbitol, B. PEG, C. Dinitrophenol, or D. Mannitol. Somatic fusion, also called as protoplast fusion, is a type of genetic modification in plants by which two distinct species of plants are fused together to form a new hybrid plant with the characteristics of both a somatic hybrid. The fusion of protoplasts from different plants to form a somatic hybrid cells and the subsequent regeneration of plants from the callus tissue produced from the proliferation of the fusion product was an extremely difficult achievement. The aim of this work is to produce hybrids which cannot be produced by normal means. However, isolated protoplasts will not aggregate and fuse easily in the absence of an inducing agent. Several chemicals have been used successfully to induce fusion, but the most successful and the most widely used at the present is polyethylene glycol, that is PEG. The effects of PEG are not specific and it will thus promote the aggregation and fusion of protoplasts from the same or different species. Here is a diagram to explain this better. PEG-induced protoplast fusion. Here the protoplasts are first brought together by aggregation by PEG. Fusion occurs during PEG dilution due to disturbances created in the plasma membrane. This forms a heterokaryon, which leads to a hybrid cell. So the correct answer is PEG, that is option B. The rhizobia synthesize specific nodulation factors, that is host features, for the recognition, which are A. Kinases, B. Flavonoids, C. Lipocyto-oligosaccharides, or D. Neutral lipids. Rhizobia elicit their specific leguminous host, the formation of new organs called nodules, in which they fix nitrogen. The rhizobia nodulation genes specify the synthesis of lipocyto-oligosaccharide signals, that is the nod factors or NFs. Each rhizobial species has a characteristic set of nodulation genes that specifies the length of the cyto-oligosaccharide backbone and the type of substitutions at both ends of the molecule, thus making the nod factors specific for a given plant host. At extremely low concentrations, purified nod factors are capable of eliciting on homologous legume hosts many of the plant's developmental responses characteristic of the bacteria themselves, including cell divisions, and triggering the plant organogenic program. Thus, the correct answer for specific nodulation factors for recognition are lipocyto-oligosaccharides. So option C is correct. The acceptors of electrons in lactate fermentation and alcohol fermentation are. For this, to understand this better, we have the diagrams of both lactic acid fermentation here as well as alcohol fermentation. In lactic acid fermentation, pyruvate is converted into lactic acid or lactate. This process requires the enzyme lactate dehydrogenase and it takes 2 NADH molecules to give 2 NAD plus H plus. In case of alcohol fermentation, pyruvate first forms acetaldehyde with the help of enzyme pyruvate decarboxylase, which removes 2 carbon dioxide units. This acetaldehyde undergoes dehydrogenation with the help of alcohol dehydrogenase to form ethanol. This process also requires 2 NADH to give 2 NAD+. So, in fermentation, the final electron acceptor for lactic acid fermentation is pyruvate, where you can see the exchange here, and for alcohol fermentation, it is acetaldehyde, which takes up 2 NADH to give 2 NAD+, or some other similar organic molecule can work in its place. So, again, just to brush it up, for lactic acid fermentation, the final electron acceptor is pyruvate, and that for alcoholic fermentation is acetaldehyde. Which of the following are called epimers? A. Sugars that differ in the placement of OH on C1. B. Sugars that differ in the placement of OH on C5. C. Sugars that differ at one stereocenter. Or D. Sugars that differ only at middle carbon. So first we must understand what are epimers. Epimers are carbohydrates which vary in one position for the placement of the OH group. The best examples are D-glucose and D-galactose. Both are monosaccharides and both are D-sugars, meaning that the OH group on the carbon 5 of these hexoses is located right in the Fischer projection. The only difference between these two is on carbon 4. For D-glucose, the OH group is on right on carbon 4, and for D-galactose, it is on the left. A single change makes D-glucose and D-galactose epimers. They are not enantiomers, they are not diastereomers, they are not isomers. They are only termed as epimers. Here is a diagram to understand better. As you can see, the fourth chiral center, the fourth carbon, that chiral center is the only one which has a change, a different handedness, if we can say that. The two isomers, different in terms of only one functional group, are thus called as epimers. These are stereoisomers to be specific. So, the correct answer is option C. Which of the following stages of glycolysis is exergonic? Now first we have to understand what exergonic means. Exergonic is any reaction which releases energy. Any biochemical reaction which releases energy is termed as an exergonic reaction. In glycolysis, there are 3 steps. The first, the third, and the 10th. These are highly exergonic reactions. There are also regulatory steps involving the enzymes hexokinase, phosphofructokinase, and pyruvate kinase. Most biological reactions, as you can see clearly in the glycolysis process, are reversible. That is, they can occur in both forward and backward directions. However, these steps, that is the first, the third, and the 10th, cannot be reversed with the help of these enzymes themselves. So, the exergonic stages of glycolysis are step 1, where glucose is converted to glucose 6-phosphate. Step 3, where fructose 6-phosphate is converted into fructose 1,6-biphosphate with the help of enzyme phosphofructokinase 1. The first step involved the enzyme hexokinase. And the last step, that is where the phosphoenol pyruvate is converted into pyruvate with the help of pyruvate kinase enzyme. Glycoproteins are carbohydrate proteins in which the carbohydrate link with the formation of carbohydrate proteins, that is glycoproteins, is called as glycosylation. Carbohydrates can be attached to proteins to form glycoproteins. In glycoproteins, the carbohydrate chains are either attached to nitrogen atom on the side chain of aspargine, that is N linkage, or to the oxygen in the side chain of serine or threonine amino acids, which is O linkage. This is O linkage where oxygen side chain of serine or threonine is attached to the carbohydrate, and this is N linkage where nitrogen side chain of aspargine is attached to the carbohydrate. So, the amino acids involved in this process can be serine or threonine in case of O glycosylation and aspargine in case of N glycosylation. Which one of the following biological phosphate molecule contains the highest bond energy? A. Phosphoenol pyruvate, B. ATP, C. 1,3-biphosphoglycerate, or D. Phosphocreatinine. Now, to understand this question, we have to know what it means to have high energy phosphate bonds. These are pyrophosphate bonds or acid anhydride linkages formed by taking phosphoric acid derivatives and dehydrating them. On hydrolysis of these compounds and breaking of these bonds, a lot of energy is released under physiological conditions. Thus, the process is exergonic. Out of the given options, all these high energy phosphate compounds, I have converted them into their respective forms after hydrolysis. So phosphoenol pyruvate on hydrolysis forms pyruvate plus inorganic phosphate, which releases an energy of minus 62 kilojoules per mole. For 1,3-BPGA, that is 1,3-biphosphoglycerate, it forms 3 PGA and inorganic phosphate, releasing minus 49 kilojoules per mole of energy. Creatinine phosphate is hydrolyzed to creatinine and inorganic phosphate, and it releases minus 43 kilojoules per mole. And for ATP, it releases minus 31 kilojoule per mole and forms ADP plus inorganic phosphate. This low energy release of ATP allows it to phosphorylate compounds which are ranked much lower than it. It is also favorable to synthesize ATP from ADP by higher ranked phosphoenol pyruvate and creatinine phosphate like compounds. If you see clearly, these four options are already arranged in the descending order. So the highest bond energy is held by phosphoenol pyruvate at minus 62 kilojoules per mole. So the correct answer is option A, that is phosphoenol pyruvate, which is the biological phosphate molecule to contain the highest bond energy. In uncompetitive inhibition, the inhibitors bind with dash, dash and dash. Which of the following options is the most appropriate to fill in the blank? A. Free enzyme decreases, no effect. B. Free enzyme increases, no effect. C. Enzyme substrate complex only decreases. Or D. Enzyme substrate complex only increases, decreases. For this, we shall refer to this diagram. Now, here, this is the enzyme. It has an inhibitory site and an active site. On addition of the substrate fits into the active site and it influences the inhibitory site. On binding of the substrate, the inhibitor site slightly changes and allows the binding of inhibitor. So the inhibitor can only bind when the enzyme substrate complex is already present. So, in uncompetitive inhibition, inhibitors bind with enzyme substrate complex only. Now, as compared to when we talk with respect to the Vmax and the Km values, both of these values see a considerable change. On addition of the inhibitor, the Vmax value, 1 by Vmax value slightly increases. So, the Vmax value decreases. On addition of the inhibitor, the Km 1 by Km value decreases. So, the value of Km also decreases. So, the correct answer here is option C. It binds to enzyme substrate complex only. The Km and Vmax values both decrease. So, the correct answer is option C. Thank you for watching. Please like, share and subscribe to vEgyan Ghar's YouTube channel for more such videos.